see my slides?
Yes, but not in presentation mode.
Not in presentation mode. Okay. I'm going to do it this way.
There you go.
Right, perfect. All right, well, thank you so much for inviting myself and Ficado to give you guys a talk. Ficado and I have had a really great partnership and collaboration for a few years now, and it actually all started with PMedic. So Karen Rodland had was talking with Ficado, or they were discussing some things that they could do, and Karen actually put Ficado in touch with me, I think about 3 years ago, I think, 2018, and since then we've worked together on like 4 or 5 different projects, and then today we're going to share one of our publications with you guys. So we're going to talk about Zika viral infections and the work we did with using lipidomics, and then about PNL, and then the subsequent work that Ficado's lab did to try and understand a little bit more of exactly what was occurring during replication in regards to lipids and lipid metabolism. So this work that we're presenting today was published last year in Nature Communications, and I just wanted to highlight the PNL team. So there's myself and June, Lisa Brammer, Kelly Stratton, so our biostatisticians. So if the OHS2 team works with PNL, you'll commonly work with our biostatisticians, who are wonderful. And also a part of the PNL team was Tom Metz, who's the head of our metabolomics group here. So just to give a little bit of background on the DoE missions, we are at a Department of Energy facility, and the missions are related to addressing critical national needs. And a few years ago, when I used to give this presentation, I used to talk about how viral infections are national importance. And I think we are all well aware now of why they are of a national importance. So that's one of the ways in which studying viruses and virus infections and the impact on host response falls within the DoE missions. And then also scientific discovery, which is a big aspect of what we do, in particular within the integrative omics group. We've done quite a bit of viral infection work and a lot of virology at PNL, again, using mass spectrometry techniques, proteomics, metabolomics, and of course, lipidomics. So again, this is an older slide, as you can see, last updated in 2019, pre-COVID, where I discussed the importance of a variety of viruses, and also just wanted to highlight how viruses are a way of life. And we all know that now. Viruses are definitely a way of life. But also to highlight every few years, or as one of our virologists at PNL, Christy Oxford, said, during everyone's presidency, they get to experience a virus outbreak that's going to impact a community that they need to be concerned about and address. And so as you can see, based on this list, I just put some of the common outbreaks that have impacted us and made global news. And you can see that every 3 or 4 years, something emerges that seems novel. So we're in 2022 almost, so who knows what's what's next, but we do know something is next. So what can we learn? How can we continue to improve and understand host response and come up with therapies or that can help, minimize the impact of these infections? So lipidomics, how can lipidomics help us understand what's going on? So we use lipidomics to help us understand identifying host factors that can help determine outcome. Also to enable us to better understand what's going on during the virus infection, including like the host response. Are there any potential ways that we can improve prevention or identify treatment options? But also lipidomics, I think of it as a, just kind of like a team player. Lipids don't work alone. They work in coordination with proteins and metabolites. We have genes and transcripts. So really, lipids is part of the entire system. And so we try to look at things together. And so you'll see with some of the work that Ficado has done, the lab-based work, how lipidomics and working obviously with proteins and stuff can come together to help us understand what's going on. So what is lipidomics? Lipidomics is the study of the structure and function of a complete set of lipids. So the number of lipids that we identify within a sample or system, we call that the lipidome. So produced in a cell or organism and their interactions with other biomolecules, again, proteins, metabolites, and things like that. So I've mentioned kind of lipids are obviously important, but they in particular, we've done a lot of viral work here, and lipids are required and critical for the infection and replication. So viruses, I think, are kind of like smart, if you want to call them organisms, but they can use a variety of different ways to get into a host system. And one of the ways I find is really fascinating is that they can trick a cell to think that they're apoptotic debris. So in this, on the left side of the screen, you can see, which is just a cartoon of an Ebola virus. And what they'll do is they'll move a certain type of phospholipid from their inner membrane to the outer membrane. And that type of phospholipid indicates to a macrophage that I'm debris, I need to be removed. And so here comes along an unsuspecting macrophage, engulfs that virus, and now that cell and subsequent system is infected. So they can use tricks using lipids to actually infect the system. And then also lipids are extremely important for replication and assembly, in particular the flaviviruses, which Zika is a part of. There's a lot of lipid remodeling that's going on, and they use these replication complexes to form. And Fakato has a really nice image of those complexes that he'll show in his portion of the talk. And then also lipids are involved in the release of viruses from the host cell, releasing it to the environment or to another neighboring cell. So lipids are required for infection, and they're involved at pretty much every single stage. So, and with that, they dramatically affect the host. Every lipidomic study we've seen here at PNNL, which has been I mentioned quite a few, the lipids are almost always changing with infection. And also, we just wanted to highlight too that lipids are dynamic and responsive to their environment. So if something is occurring, the lipids are going to respond to help the host or the system respond to that environment, and to just figure out how can we do things. So this is an example here of if you bring material into the endocytic pathway, as material is transported through all these different endosomes to the lysosomes, you can see over here on the right side of different types of lipids, such as phospholipids or sphingomyelins, and you'll hear about that later. It's a type of sphingolipid, or cholesterol. You can see how they go either decrease in abundance, or they increase in abundance, or they come in and out as you're transporting material through this particular pathway. So we've mentioned lipids are required. They're really important for viral infection and replication. They're responsive to their environment. And also lipids are abundant, but they're also they're structurally diverse. So we traditionally identify around 400 lipids within a system that we're analyzing. It could be a little more, it could be a little less. But just like organisms, lipids have different types of categories. So when you hear some of these later on, you get a little bit of an understanding of where they're coming from. So there's 8 different types of main lipids. You can kind of think of this as like the kingdom. And then with each one of these, there's these main classes. So if we look at phospholipids, there's 20 plus different types of phospholipids. One of those being phosphatidylcholine. It's a common one that we see in mammalian systems. And then with each one of these main classes, then we have a bunch of different subclasses. For the one that most people are familiar with is when you've seen ever since like high school biology, college, you have your phospholipid here with 2 fatty acid chains, and this over here, part of the head group is the choline. So this is, very traditional lipid. Other lipids that fall in this subclass have a single chain, or they have different linkages, but basically the whole point of this is that lipids are very diverse. The fatty acid chains can be from 10 carbons up to 24 carbons, which is more common in mammalian systems, and then you can have no double bonds, or you can have upwards of 6 double bonds, which is, a big portion of why lipids are so structurally diverse. So to go into why we're meeting today, a little bit of a background on lipids, but to talk about the Zika virus. As I mentioned, it's an emerging virus from a few years ago. It's a flavivirus. Zika is an envelope positive sense single-strand RNA virus that was discovered in 1947 in Uganda. And then from there, it's kind of emerged in like smaller populations with just a few people being affected, just like a little bit around the globe. And then, as we all know, 2015, 2016, there's just an explosion of Zika virus infections. And what really hit home, I think, to many people was the association of that infection with severe birth defects. So although I know a lot was learned quickly with that outbreak, there was still quite a bit that remained poorly understood. And so that's when Ficado and I started working together. As I mentioned, Karen Rodling got us in touch, and he had these systems where he wanted to better understand what was going on with lipid metabolism, and that's when we started working together. So here's our traditional workflow for lipidomics. We always meet with the collaborator. So Ficado and I met, and he discussed what he wanted to do, and we talked about study design. So usually a lot of times when you work with us, we'll bring in a biostatistician to make sure that your sample set has enough power to make sure that what we do addresses your scientific question. We extract the lipids, and I'll talk about that a little bit. Our standard platform is using liquid chromatography, tandem mass spectrometry, and I'll tell you why we find that to be a really useful platform. We identify the lipids and have an associated abundance value with that. We do statistical analysis, and then we move on to the interpretation. So here was this study design. So we started, Ficado's lab started out with HUH7 cells that were mock infected, or pretend infection, and then also Zika infected with an N of 5. So there were 5 replicates. We also did more cellular relevant cells, so neural cells, which Ficado will talk about in a few slides from now. So we did these HUH cells. We also did some neural cells, mock and infected. We extracted the lipids at 2 different time points, so 24 hours post-infected and 48 hours post-infected, and then we did our global profiling. So that's the general study design and outflow, but to take a couple steps back to show you exactly what we did. For sample prep, we use a very traditional method. It was developed in the 1950s, called the Folch extraction at PNL. So the Folch extraction, what it does is it separates metabolites and proteins and lipids. So at PNL, we actually can use each one of these layers. And so this is a method that we call MPLEX for metabolite protein lipid extraction. And if you use a particular ratio of these solvents, so 8:4:3, it automatically causes a phase separation of all these materials. And so the lipids, almost all the lipids go down into this layer. So this method is great for global metabolomics. It's really beneficial if you want to look at different omics materials within the same sample, which is really beneficial, decreases variability, but also if you don't have much material, we can do quite a few things with one sample. And then also for a lot of the pathogen work we do, the great thing about this method is because we use chloroform, it also inactivates the pathogen at the same time. So as Ficado can attest, and quite a few of our partners can attest to, and collaborators, that prior to shipping samples to PNL, we want to make sure that the samples are inactivated, especially if they're coming from a high level of bio-level safety. So this is a paper that we published in 2017 showing and confirming that with a variety of different virus infections, this method actually inactivates the virus. So if you were to collect a sample from the metabolite layer, protein layer, lipid layer, the virus has been inactivated. But we have to be cautious if that particular virus does not have a lipid layer or lipid associated with it, but then it's much harder to inactivate. But with Zika, it has lipids, it was inactivated, and we were able to use this procedure to extract the lipids and activate them. So Ficato's lab actually did that, and they sent them to us to be analyzed. So as I mentioned, we do LC tandem mass spectrometry. So we separate the total lipid extract that we've generated from that MPlex or Folch extraction. We separate over a 34-minute gradient, and that spreads out the lipids. So we can decrease, or I guess increase, our ability to ionize them and detect a variety of different type of lipids, from lower abundance to the ones that are more highly abundant. And as you can see here, we detect a variety of different types of things, including phospholipids with 1 chain, 2 chains. We can get a variety of different types of sphingolipids, which are important for this particular study, such as sphingomyelins and ceramides, and also triglycerides. We identify the lipids, so we use an in-house software that we developed and published also a few years ago. And what this software allows us to do, it allows us to identify the type of lipid that's present. So in this case, it's a phosphatidylcholine, and then it also allows us to identify the fatty acids that are associated with it. So we look at the fragments that, again, allow us to identify the structure of the lipid. In this case, it's a phosphatidylcholine with 16 carbons and 0 double bonds highlighted here. And the second chain is 18 carbons and 2 double bonds highlighted here. So this is just when you see further on lipids annotated, you understand exactly what this annotation means. And then from there, Lisa Brammer and Kelly Stratton did a statistical analysis. And this is just a very quick overview of what we found. Over 330 unique lipids were identified. You can see here in this PCA plot that the mock infected separated really well from the viral infected, and then the 24-hour time point separated from the 48-hour time point. And here's just a bubble plot showing all the lipids that were identified at 24 hours and 48 hours. And you can see there's quite a bit that's changing and statistically significant. A lot of it is statistically significant. But one particular type of lipid that caught the interest of Ficato and his team were the sphingolipids. So the ceramides, which you can see here in these blue bubbles, and the sphingomyelins, which you can see here on these light blue bubbles. So from there, I will pass this over to Ficato so you guys can see what he did with this data and the follow-up lab work that they've done.
All right. Can you see my slide?
Yep, we can see it. It's perfect.
Wonderful. Okay, Jennifer, thank you so much for that nice background. That's helped a lot. So as you have seen, we did that global lipidomics to identify, or to just basically to map the lipid profiling of infected versus healthy cells. And then what we see is that Zika infection remodels the lipidome of the host cells, and then there is some data that we found. For example, triglycerides decreased and diacylglycerides increased, which is consistent with previous report that shows dengue virus infection actually remodels the neutral lipids, especially the triglycerides and the diacylglycerides. And then the next question for us was that which viral factors is responsible for remodeling those lipid pathways. So just to remind you that Zika virus is a positive sense RNA, and it has around 11 kilobase per genome size, and it has a total of 10 proteins, 3 structural, which are the capsid, membrane, and the envelope, and 7 non-structural proteins. Here in this picture, you see the NS1, NS2A, and so on and so on. So the three structural proteins are usually involved in forming the virus itself, and they are responsible for entry and the membrane fusion during infection. And the non-structural proteins are known to be involved in endoplasmic reticulum remodeling and forming these replication factories. And then they are responsible for replication in general. Although, despite their limited number of proteins, the virus only have 10 proteins, but the function of most of these non-structural proteins is actually not known. So it was quite difficult really to determine which lipids, which non-structural proteins are important. While the enigmatic nature of the Zika non-structural proteins present a challenge to defining the lipidomic, defining the mechanistic basis for our lipidomic results, two lines of evidence, maybe three, led us to investigate the role of non-structural 4B proteins in remodeling of the lipids. This is mainly just to give you a little bit about the replication complexes that is formed within the endoplasmic reticulum. Once it's said that the virus gets in, it actually remodels the endoplasmic reticulums, and it forms this structure called convoluted membranes that burst from the endoplasmic reticulum. And then this convoluted membranes is mainly driven by the non-structural proteins. And one of the non-structural protein that is really involved in this is NS4B. And there is one study a few years ago in dengue virus where they individually expressed the different non-structural and structural proteins and measured the ER stress and the autophagic processes. And what they found was that NS4B, which is this transmembrane proteins, is this gives the strongest causes the strongest ER stress, and also it induces the strongest autophagy pathways. So which means that both cellular processes are dependent on lipid metabolism. And also there is one paper that shows that the hepatitis C and hepatitis C virus, which is also in Flaviridae family, just expressing NS4B of hepatitis C virus causes lipid remodeling. Because of that, we decided just to perform another lipidomic where we transfected NS4B and then also as a control empty vector. And we send the samples to Jennifer to basically do exactly the same type of lipidomic analysis to see if there is any similarity between virus infection and NS4B transfection. What we see from the get-go is that there is nice separation between empty vector, which is our control, and NS4B transfected cells. And then when we actually do this careful analysis, what we see is that, which is actually just it nicely mirrors the virus-infected lipidomics. So, but which is actually very particular in that, this is a bubble plot. On this one, this is a log two-fold change, and then the size of the bubble corresponds to the p-value. The larger the circle, the more significant it is. And then what is really striking is that we see a very distinct change within the sphingolipid species. Okay, there are changes in other species, but if you look at the changes within the sphingolipid pathways, is very stark. Ceramides are highly enriched, and the sphingomyelins are reduced. So one way of looking at this is that what are the lipids that are enriched in infected cells and NS4B transfected cells. When we do this, when we do this correlation, actually the observation is quite stark and very, very clear. What we find is there is a significant enrichment of ceramide species, one of the sphingolipids, in the both infected NS4B cells and the NS4B transfected cells, which shows that these lipids might be important for Zika virus infection. So how are we going to test this? Just a little bit about the sphingolipid pathway. The sphingolipid pathway starts in the endoplasmic reticulum by the transfer of serine onto palmitoyl-CoA by enzyme called serine palmitoyltransferase (SPT), which produces dihydrosphinganin, and then forms which is important for ceramide. Ceramide is then converted into sphingomyelin or glucosylceramide, which can be converted further by different enzymes to higher glycosphingolipids. So this is just the ceramide contains sphingosine and fatty acids, and depending on the head group, if the head group is choline, it is a sphingomyelin. If the head group is glucose, it could be glucosylceramide, and so on and so on. Luckily in the field, there are two important small molecules that block enzymes within this pathway, myriocin and FEB1. Myriocin blocks the very first step in this pathway, serine palmitoyltransferase, and basically blocks the whole sphingolipid biosynthesis. And we have another enzyme called feminicin B1 that blocks the activity of ceramide synthase, and again, and then it blocks ceramide. So to test the inhibitory effect of these molecules, what we did is we labeled the cells, we treated the cells with these compounds, and labeled the cells with 14C labeled serine, which is the very, very first precursor. And then we can look at the formation of different species of the sphingolipids. In this case, it's sphingomyelin. This is the thin layer chromatography showing that the sphingomyelin in myriocin and FEB1 treated cells is completely absent, showing the efficacy of these molecules. So what we did is we treated Q7 cells with myriocin and FEB1, and we have also vehicle control. And then we infected cells with Zika virus. And after different time point, 24, 48, and 72 hours, we collected the supernatant and quantified the amount of virus that exists in inhibitor-treated and control cells by plaque assay. This is just to show you that myriocin treatment and FEB1 treatment significantly reduces the viral shedding into the media, showing that this sphingolipids, this pathway is essential for Zika virus infection. And this is another way of looking at the same data, where instead of quantifying the amount of virus using plaque assay here, we just actually quantified the genome of the virus in the supernatant by RT-PCR. And again, showing myriocin and FEB1 treated cells have significantly less amount of viral genome as compared to the control. And then obviously we want to confirm this by genetic knockout, and just to get rid of any off-target effect of the small molecules. We did have some knockout cell lines in our lab at the time. These are HAP1 cells and the dendritic cells, DC2.4 cells that lack SPTLC2. This is a subunit of serum palmitoyltransferase, which is a target of the myriocin inhibitor. So cells that lack serum palmitoyltransferase are resistant to the virus as compared to controls in both cell lines. Again, showing that sphingolipids are important in for Zika virus infection. And obviously, we know that Zika virus is known to be a neurotropic and mainly infects neuronal cells within the central nervous system to cause microcephaly. That's how actually it causes. And the studies have shown that human neuronal progenitor cells can be used as a proxy for physiologically relevant primary cells. So that is what we decided to do, because the previous studies are not neuronal cells. In this case, we just used, we decided to use NPCs and also neoblastoma cell lines, SHSY5Y cells. And again, we used these two inhibitors, myriocin and FB1, and quantified the amount of virus that is released into the media after different time points. And actually, in these primary cells, the impact of these inhibitors upon the viral shedding is even more significant as compared to the other cell types that we have seen, showing that again, this the critical role of sphingolipids in Zika virus infection. So okay, the sphingolipids are, we know that sphingolipids are important, but we didn't know which step they are relevant. Is that in the entry or viral replication or maturation or release? We have done a lot of experiments to basically define the step at which sphingolipids are required. So one of the experiments to look at the entry and or membrane fusion step is that we performed plaque assays where we did infection and we quantified the amount of virus that retain in the cells, gets in the cells after 10, 20, and 45 minutes, because that actually, that is the time where the first step is. We didn't see any difference between myriocin or FEV1 treated cells as compared to the vehicle. If anything, treatment cells that were treated with FEV1 seems to have actually high level of variance, showing that might be FEV1 might actually increases infectivity. And then to look at the replication step, we know that Zika virus replication starts at 4 hours. And then what we did is we quantified the amount of viral RNA within the cells after 4, 8, 12, and so on time point. And what we determined is that in myriocin and FEV1 treated cells, we have a significant reduction in viral genome, showing that these lipids are important not in the entry but in the replication stage. And the next step is that obviously which sphingolipid species is important, because we know that at least myriocin and FEV1 treatment is required. At least we know that ceramide production is important, but ceramide can be converted into sphingomyelin or glucosylceramide, galactosylceramide, or even higher glycosphingolipid like GM1, GM2, and other gangliosides. So we want to determine which sphingolipid species is required for this. Again, for this one, we went with the first thing we did was that we tested the infectivity of cells that lack sphingomyelin synthase that converts ceramide into sphingomyelin. And what we saw with that is actually very very striking and unexpected in a way. So the one that lack sphingomyelin synthase become extremely permissive, up to 100-fold more sensitive to the virus as compared to the wild type. And when you put back the gene, they become like a wild type, showing that a sphingomyelin synthase deficient cells are sensitive to this one, which means that it is not the sphingomyelin that is important for replication. It is the ceramide that is important. And then to further verify that, what we did is, so ceramide in the cells are, in addition to the sphingomyelin salvage pathway, there are other mechanisms. One is the endogenous pathway in the which FEB1 is involved, and then the myriocin. And the other one is the salvage pathway, and which means that the sphingomyelin can be degraded back to ceramide. So we got this inhibitor that blocks sphingomyelinase, and when you treat cells with sphingomyelinase inhibitor, you get a significant reduction in viral shedding. And then when you exogenously add a recombinant sphingomyelinase into the cells, that basically accelerate the production of ceramide by degrading sphingomyelin, you actually enhance the virus production. Again, showing that ceramide is the lipid that is important for replication. And then again, just to confirm this, what we did was that we had those sphingomyelin synthase knockout. When you treat the cells with sphingomyelinase inhibitors, you don't really see that reduction because the cells are actually, they don't have much sphingomyelin. So adding sphingomyelinase didn't really make much difference. But those cells that are reconstituted, those cells that have sphingomyelin, treated with these inhibitors become permissive to the cells, to the virus, again, showing that again, ceramide is important for Zika virus replication. And this is just to show you that whenever the cells have higher level of ceramide, it's become very pro-viral. The virus like that replicates faster. And then whatever, whether that is this genetic knockout or small molecules that prevent accumulation of ceramide tends to block infection. And then we know that in mammalian cells, again, we have different types of ceramides that are catalyzed by different ceramide synthesis. In mammalian cells, that we have 6 of them. Some produces short chain, some produces long chain ceramides. Then we want to know which ceramides are important for these processes. So we went back to our lipidomic data, and what we did was that we looked at the level of ceramide, the short chain, the D18 and 16 acyl chain species versus the long chain, for example, the 24 carbon species. What we see is that long chain ceramide that is catalyzed by mostly ceramide synthase 5 and the ceramide synthase 6 is highly enriched in Zika virus infected cells, both at 24-hour and a 48-hour point. So obviously, this brings us the question how ceramides are actually important for Zika virus replication. So to do this, we went on and then did some careful analysis by high-resolution confocal microscopy. We have done a lot of experiment with that, but the key message is that ceramide actually redistribute to the serum replication site. Here are cells that were infected with Zika virus, and we used NS4B antibody. NS4B is a marker for replication sites. And also we used antibody against ceramide, and we see this very nice co-localization of ceramide with NS4B, which is the replication site marker. And if you do the same type of co-localization with sphingomyelin, it doesn't co-localize. And Zika virus, ceramide. If you look at these other structural proteins of the virus, which are not a marker for replication site, for example, the envelope protein of ZIKV, it doesn't co-localize again, showing that a ceramide is specifically targeted into the replication sites. So this brings me, I think, to my conclusion, which is I think we, with this collaborative work, I've shown for the first time a detailed map of a lipidomic map of cells that are infected with Zika virus. An expression of Zika virus NS4B enriched host sphingolipids, which is kind of, we think that NS4B is the prime candidate for responsible for remodeling the landscape of the host lipidome. And then we also shown that sphingolipids are required for Zika virus replication. And we think that the ceramide sphingomyelin network is a key determinant of Zika virus infection. And I think which is really interesting is that, to me at least, the fact that ceramide is redirected into the replication membranes to form those replication factors is still, I think it's quite a finding. And we hope in the future to follow up to further investigate how small chain ceramides actually responsible for forming this replication factories. So with this, I would like to acknowledge this is, again, it is a very collaborative work. Most of the work in our lab is done by Hans Leyer, who just recently left the lab for its MD PhD studies. And we collaborated with a lot of labs within OHSU, and obviously from PNNL. Especially, I would like to thank Jennifer, Lisa, and then June as well, and Kelly and Thomas for this wonderful work. And I think I just want to really mention that we wouldn't have done this work without their help. And I think this project kind of become a starting point for follow-up studies. We have a lot of other projects are also going on as we speak. We have a couple of, I think, papers that are under review, which hopefully will be able to present in the future. So with this, I would like to end my presentation. And if you have any question, please let me know.
Thank you, Fakadu and Jennifer. That was an excellent talk. Very, very interesting. We have one question so far in chat that I will go ahead and pass along. Mechanistically, how do you think NS4B may be involved in remodeling the lipidome?
I don't know. I think the answer is we don't really know. That is something that we are plan to actually further investigate. I mean, I have a couple of hypotheses. One is that it could be that ceramide primarily is synthesized in the endoplasmic reticulum. And then once it is synthesized in the endoplasmic reticulum, it gets transported to the Golgi to be converted into a higher glycosed sphingolipid like sphingomyelin or glucosylceramides and so on. My guess is somehow during infection it gets trapped into the within the endoplasmic reticulum. Maybe NS4B somehow hijacked that transport mechanism to get into the ER. And then we know that even though I don't really have a direct evidence of this, but we know that ceramides, especially small short chain ceramides, are important for remodeling the curvature of the membranes that are important just to forming those small vesicles. Again, I think these are all testable hypotheses, and we hope to do this in the future. I don't know, Jennifer, if you have any other comment. Okay, she said she doesn't have.
Yeah, I think that's the best that you can get at the moment, but I do hope you get to test all those hypotheses. Another question, does this apply to other flaviviruses as well, do you believe?
Yes. I think we didn't really do lipidomics in this one, but we have done experiments where we treated cells with meriocin and FEV1 cells and infected with dengue virus, and we see the same type of inhibition. So from those experiments, I strongly believe that this is a general phenomenon within the flaviviruses.
Great. Next question. Is there any discussion on using the inhibitors or modified synthetic equivalents therapeutically?
This is very important, a very, I think, relevant question, obviously. We actually tried with Alahesh to treat mice with FB1 and then infect with Zika virus. That experiment became very complicated because FEB1 is toxic to the mice as well. So we ended up not, I think that study was not really very conclusive. But we could have used meriocin. I don't know why we didn't do that. But if I were to repeat that experiment, we know that meriocin is not toxic to animals. People have done animal work. Probably we could do those type of experiments. But again, I think this shows a clear role for sphingolipids in Zika infection, but its effect on mice or animals remain to be determined.
Anybody want to raise a hand and ask a question? Otherwise, that's all the questions. Oh, here's another one. Does Zika infection upregulate ceramide synthase?
Yeah, I think that is very, very good question as well. We haven't looked at that. Yeah, we didn't look at. I think that is very important question. Again, it's part of the future plan. And what is known is that NS4B, we have data that shows NS4B interact physically associated with serum palmitoyltransferase SPTLC2. That finding is not published, but we know that at least there is a physical. If we do immunoprecipitation, serum palmitoyltransferase comes down with NS4B. This tells me that there is definitely a direct regulation of this pathway by this viral proteins. Whether that eventually that interaction affect the expression of the different ceramide synthase enzymes or their transcript or the proteins, I don't know. We haven't done that. I think that's something that we would like to obviously follow up.
Great. Well, we've used up almost a whole hour. Again, I want to thank you both. As we mentioned, this is one of the first introductions that was made through this, having a team on both sides trying to generate new collaborations. And this has been sort of a seminal project I've been able to point to over and over, sort of the great work of putting these interdisciplinary teams together. So if you have further questions for Jennifer or Foucault, you can reach out to them directly. Or as I mentioned, you're welcome to reach out to me or Laura or Liz. And if you have other ideas outside this project. So thank you everyone for attending today.
Thank you.
Thank you.